So let me introduce Corey, who's part of our Speak team, and I didn't get a chance to introduce Steve Black. Both are longtime vaccinologists. Corey, let me have you take the lead on the next session.
Great, thank you. This part of the discussion is going to be talking about immunological markers and some of the more clinical aspects that we haven't touched on yet in our animal model discussion, which was really very helpful. Our first speaker is Dr. Baric, who is a professor in the Department of Epidemiology at UNC. He comes there via microbiology and virology work and has spent a good part of his career studying coronaviruses. He has already, in question and answers, talked some about the data that he'll be presenting today, but this will be a very good review, and he has some very good insights, I think, for the discussion we're about to have. Dr. Baric.
Can everyone hear me?
Yes, thank you.
I can't see any slides.
Oh, that's a problem.
Can
do you see
anyone
the
else see
subject
my
slide?
slides? Ah, here we go. Okay, so I see my slides now.
Great.
So I'll be talking about structure and function of the coronavirus spike glycoprotein. I just wanted to remind everybody what the phylogenetic tree of coronaviruses look like, which is in the middle of the lower part of the slide. Next slide. So to begin to introduce the topic of the spike glycoprotein structure and function, I just want to remind the group that there's been a long history of emerging coronaviruses over the past 800 years. The emergence of human coronaviruses and animal coronaviruses seem to be accelerating in the 21st century, starting with SARS coronavirus about 16 years ago, which is on the bottom of the slide. I don't think I have a pointer. MERS coronavirus about 7 years ago at 2012. There's a porcine coronavirus called SARS coronavirus about 2 years ago that emerged from China, and SARS-2 about three months ago. Next slide. So an important thing to consider when developing vaccines or immunotherapeutics is that there have been 6 emerging coronaviruses in the 21st century, 3 in swine populations, which I mostly did not highlight, and 3 in human. And this accelerated cross-species movement is distinct from historic trends. And so this may or may not be related to potential changes in the global ecology that regulates coronavirus emergence. But I think that we as the community need to think about this as a problem that may well likely increase or continue at the same rate over the next several decades. Next slide. The drivers of variation in the spike glycoprotein are summarized on this slide. Coronaviruses have a mutation rate of about 10 to the minus 6. This is from its RNA-dependent RNA polymerase, which has a higher mutation rate. However, there's a regulated fidelity complex mediated in part by NSP14, which is a replicase protein that interfaces with other non-structural proteins to actually repair 3' end mismatches from growing nascent strands. This is probably a regulated fidelity that can occur, that changes its performance either as a function of time post-infection or as the virus moves into altering or different host environments. The second major driver of rapid evolution in the spike is the high rates of RNA recombination. They can, in a mixed infection and culture, 25% of the progeny can be recombinant viruses. And in addition, you can have a phenomenon called modular evolution, where whole genes are swapped between strains, or modules within a protein like the spike glycoprotein, like the NTD domain or the RBD domain of the virus spike glycoprotein, that can be exchanged between strains, allowing for rapid evolution in as these viruses replicate in close high-density populations in bat species. Finally, the spike glycoprotein itself, which is shown here with the blue arrow pointing at the little blue, I guess that looks like kind of like a mushroom. The spike itself tolerates very high rates of mutation. There have been large deletions and insertions that have been introduced into the spike retaining function, although it has major impacts on tropism and antigenicity. And also spike regulates, of course, host range, tissue tropism, and transmissibility. Next slide, please. So when you think about the SARS group 2B SARS-like coronaviruses, this is the state of the phylogenetic tree prior to December 2019. The strains highlighted in yellow are SARS coronavirus and SARS-related strains that emerged either during the early, middle, or late phase of the epidemic, as well as strains that circulated in civets and raccoon dogs. The Urbani strain has the arrow pointed on it in that slide. In addition, over the next 17 years after the epidemic, researchers in China, Europe, Asia, and the United States have identified a large number of additional SARS-like bat coronaviruses, which are highlighted in pink. They fall in general into two flavors: the light pink strains, which I'll define as low risk, and I'll show you why shortly, and then the dark pink strains, which we define as high risk, and I'll show the reason for that in the next slide. So this was the current state of our knowledge of spike glycoprotein variation within the group 2B coronaviruses prior to December 2019. Next slide. So one of the experiments that we did in our lab was to ask what the pre-epidemic potential of bat SARS-like coronaviruses might be in human populations. And so we looked at the phylogenetic tree and identified strains that were closely related to the SARS-like strains, which are shown in the black box with 2003 written above it, and then synthesized the spikes of the gene shown here in red and dropped them into a SARS coronavirus molecular clone, replacing the Urbani spike. And when you do those experiments, the three spike glycoproteins from WIV1, 16, and SHCO14 could program efficient replication of primary human airway epithelial cells. These are the cultures in the far right bottom. They could use human receptors as well as SARS coronavirus. They could replicate to equivalent titers in HAE cultures. And if you synthesize the full-length genomes, you could recover these full-length viruses. The two strains that are in brackets, bat coronavirus 279 and HKU3, are lower risk strains because when you drop those spikes in, you could not recover those viruses. Next slide. So the current knowledge about high and low risk SARS-like bad coronavirus is sort of summarized in this encyclopedia of high-risk strains. The strains that had up to about 10% variation were known in bat populations. This was done by our group as well as Zhengli Shi at Institute of Virology in Wuhan. Among these, SHCO14 was the most variable at about 10%. The 2019 human coronavirus strain is about 22% variation in the spike, and all of these viruses have similar high-risk features. They use human ACE2 for entry. They grow in primary human airway cells quite well. They cause acute respiratory distress syndrome in humans as the major endpoint disease phenotype. They display age-related disease severity either in mice and humans. So in mice, if they're aged past 20 weeks, the animals progressively get more susceptible to disease, and the LD50 can drop to from 10 to the 6th to 10 to the 2nd. They also, as you begin to pass SHCO14 here at 10% variation, they escape existing immunotherapeutics, and I'll show that to you later. So an important thing to remember when we talk about vaccines against the SARS-2 coronavirus is that we already do have a broad platform of related SARS group 2B strains that exist that we can ask about vaccine performance for the 2019 strains in terms of how well they protect against progenitor strains that exist in nature. And this may reveal potential immune pathologic features that might be associated with the vaccine because of heterologous challenge. I think this is also another important point to raise because it does highlight the possibility that any vaccines that we put into people have to be considered the long-term impact of what happens if there's a new emerging SARS-like group 2B coronavirus in the future. And I'll just say that we believe that variation among these viruses can approach 35%, and they can still have contain all of these high-risk features that are shown on the right side of the slide. And so keep that in mind. Next slide. I just want to also reiterate that this characteristic is also replicated in the group 2C MERS-like coronaviruses. We know very few, we've identified a much less number of strains that are high risk, but we have identified 2. One is MERS coronavirus, obviously, and then Zhengli Shi's group has identified MERS 422. It is 35% variant in its spike glycoprotein. It can still use the human DPP4 receptor, and studies in our lab show that it can grow efficiently in primary human airway epithelial cells, in fact, as well as the MERS coronavirus. So keep this in mind. There are heterologous challenge strains that exist. All of the strains that I showed in the last 2 slides, we have animal models for, and we have reagents to look at specific neutralizing responses to each of those serotype variants. Next slide. So when you think about emerging coronavirus preparedness in terms of spike glycoproteins, I think you have to, again, recognize this large reservoir of SARS-like and MERS-like coronaviruses that are poised for emergence, that the variation can approach 35%. And so as we develop vaccines, there's really 2 priorities. First is the immediate priority of the SARS-2 vaccine formulation. But then the second is broad-based vaccines that would protect against the future emerging strains that will or are destined to emerge in the future. So please keep that in mind. Next slide. So this was, again, the phylogenetic tree in terms of the state of the knowledge prior to December 2019. In 2019, next slide, SARS-2 was identified in Wuhan, China. It is a unique clade within the group 2B SARS-like coronaviruses. It is about 22% different as compared to the SARS and the low and high risk SARS-like strains that have been identified earlier. That corresponds to about well over 6,000 nucleotides across the 30,000 nucleotide genome. Its closest relative at about 96% identity is RATG13, which differs about 1,200 nucleotides. Next slide. The genome organization of Wuhan and SARS-2 are the same, and we're going to focus on the spike glycoprotein shown there in the big rectangle with S written in the middle of it, which is the location of the spike gene. Early modeling studies by our groups and others had argued that the SARS-2 spike glycoprotein would be able to bind to the human ACE2 receptor. This is some modeling data from that paper. Green is the ACE2 molecule, purple is the receptor binding domain of SARS-2, and it looked like it made a very nice interface, and that's been now demonstrated by several groups using biochemistry assays, as well as a capacity to infect cells overexpressing the human ACE2 receptor. Next slide. In addition, Yan et al. Recently published the cryo-EM structure of the SARS receptor binding domain, the SARS-2 receptor binding domain bound to the human ACE2 molecule. And I'm not going to go through this slide, except to note that the interface mediated by the SARS-2 strain is very interactive and forms a very tight integrated network to promote docking and entry into cells. Next slide. You can compare the receptor binding domains across the high and low risk SARS-like group 2B coronaviruses. These are all aligned to the SARS coronavirus sequence at the top. There are 14 contact interface sites. And as you move to more antigenically distinct strains from SARS coronavirus down to SHC014, which is shown in the red box, you can see that as the darker green colors change to lighter green or yellow, the resiliency of that residue to interface with the ACE2 receptor is dropping slightly. and as it turns red, it is clashing with the ACE2 receptor interface. SHC014 and all of these strains in the top part of the box can use the human, civet, bat, and mouse ACE2 receptor quite well, even when 7 of 14 of the contact interface residues are different, which is shown in SHC014. The Wuhan strain retains 8 of 14 contact interface sites, either at high or slightly lower level of efficiency of interaction, and also has unique variation that has not been seen in other strains. Note that the low-risk strains contain deletions within the receptor binding domain, which sort of explain why these viruses can't use ACE2 to mediate docking and entry into cells. Next slide. Work in Barney Graham's lab and other laboratories have identified at least 3 major sites or antigenic sites on the surface of the spike glycoprotein. This is at the top of the slide is a linear representation of the structure of the spike glycoprotein, which is divided into an S1 and an S2 phase. There are specific cleavage sites that are unique to each of those strains that help mediate docking and entry. Unique in the SARS-2 glycoprotein is a furin cleavage site that's not seen in any of the other SARS-like strains. In addition, TMPRSS2 cleavage sites have been identified. Most likely there are other cathepsin and other potential cleavage sites that could be critical for programming conformational changes in the spike glycoprotein that are needed post-receptor binding to expose the fusion domain, shown here as FP, to fuse the glycoprotein to the plasma membrane to mediate virus entry into the cell. In terms of vaccines, the 3 major antigenic sites, again, are RBD, NTD, and in the S2 region. And in the next slide, I've annotated, next slide. Oh, one slide, yeah. And next slide, I show the amount of the accumulating variation shown by green dots superimposed onto the SARS coronavirus spike glycoprotein structure. The SARS 2003-2004 outbreak strains are shown in the red box on the left. Even civet and raccoon dog strains represented by SZ61 and 03, that should be HCSC6103, not 13. Even that small amount of variation in that civet strain can mediate escape from vaccine induced neutralizing antibody responses, both in young and especially in aged animals, to mediate a strong Th2 immune pathology. So it doesn't take much variation for natural variants of SARS-2 or any related SARS strain to escape immune-based neutralizing titers, especially in individuals who mount sub or lower level of responses. As we move into the bat coronavirus strains, you can see there's significantly more variation. SHC014 has about 10%, followed by SARS-2, which has extensive variation across both the surface exposed top of the glycoprotein and the RBD and the NTD domains, as well as in the conserved stock regions. And so the big question, of course, is as you move across this antigenic variation landscape, what's the impact on immune therapeutics and vaccines? Next slide. In addition, coronavirus, next, go back one more slide. One more. Oh no, this is good, this is good. Sorry. This was published by Walls just recently in Cell, who identified the glycan shield that exists on SARS coronavirus, SARS-2 spike glycoproteins. These are shown by the little blue balls that are on the RBD surface exposed binding domains at the top of the molecule, as well as in the NTD and the S2 domains of the protein. Now, if you take all of the variation that I showed on the last slide and annotate it in terms of where conserved regions are on the spike glycoprotein, that's shown on the lower part of this slide. The S2 portion forming the stock of the trimer has conserved sites that potentially could induce broad-based vaccines against all coronaviruses. Next slide. We have looked at a large panel of most of the monoclonal antibodies that were raised against SARS coronavirus, unfortunately, were panned using receptor binding domain regions. And so, in essence, the antigenic map of SARS had never been completed. I guess the person who's probably the closest to resolving that knowledge gap is probably Barney Graham's group at the NIH VRC. We worked extensively early on with Antonio Lancevecchia, Wayne Marasco, and George Giorgio, who made a variety of monoclonal antibodies to the SARS receptor binding domain. Among those, about 35 antibodies, of which a subset are shown here, 4 actually had high potency to all these SARS-like strains through the civet strains, as well as WIV1 and WIV16, which were bat strains. And these are the antibodies 109.8, 215.17, 230.15, and 227. All of those antibodies could elicit escape mutations quite easily in the receptor binding domain, and the changes are shown across the top of the x-axis on this slide. In some cases, antibodies selected for mutations both within the receptor binding domain and in the S2 domain. The 230 antibody was one of the most potent antibodies shown here with the blue arrow. The structure of that was solved in Wall et al. That was published in Cell about a year ago, I think. And 230 binds to the top of the receptor binding domain shown here. If you take the 230 antibody, as well as these other highly broadly cross-neutralizing antibodies, and look in the lower left-hand corner of the plot, FM6 is quite potent against Urbani, but it does not neutralize SHC014. 230 neutralizes Urbani, but not SHC014. 227 Urbani and barely achieves a 50% neutralization of SHC014 at the highest concentration of antibody, and 109 shows a similar phenotype. When you vaccinate animals with the SHC014 spike glycoprotein, which again is 10% different, you see about an 8-fold reduction in the neutralization titers to SARS coronavirus, which is shown on this slide, and this was published sometime earlier. Next slide. So some key priorities, I think, certainly are to characterize the SARS coronavirus 2 neutralizing epitope map, identify broadly cross-reactive neutralizing epitopes so that we might be able to focus vaccines on specific targets, identify putative enhancing epitopes if they exist, and I would have to argue that if they didn't potentiate disease in vivo, the interest level in this as a research topic should go down. Stan Perlman has identified T-cell epitopes in SARS and in mice, but we probably need to do more broad-based screening of infected individuals to get a much more precise map of the T-cell epitopes that span the SARS-2 genome, proteome, and of course determine key correlates of protective immunity. Next slide. I want to jump back to this SARS vaccine complication slide in relation to spike for just 1 second. In animal models, vaccine efficacy in aged populations, that's 1 year or older animals, can be reduced in terms of the neutralizing titer by 8 to 10-fold. If you use a heterologous challenge strain, you can get an 8 to 10-fold reduction in the neutralizing response as well. And so when you put those two together in an aged animal, it can become quite deleterious to the performance of a vaccine. The final thing that I want to mention is that the concept of outbred populations and how animal models, genetic reference populations may be of value to identify potential vaccine complications. Next slide. This is an experiment we did in a 8x8 diallele using the 8 founders of the collaborative cross. And so an F1 population was shown. The sire is the different 8 CC founder mice strains that include 5 domesticus strains, and then a wild-derived domesticus, a musculus, and a castanea strain as F, G, and H. The sires at the top, the dams are at the bottom. This is not a VRP spike vaccine. It's the SARS doubly inactivated vaccine. It was administered to these animals at 12 months. They were boosted at 13 months and then challenged at 15 months. And in contrast to what's seen in regular BALP-C or B6 animals, either young or old, where you do see immune pathology but not increased lethality or weight loss, these phenotypes begin to emerge when you scramble the genetics of the population. So for example, anything here in blue is an animal, is a F1 group of animals that had a beneficial response following vaccination, and that the vaccine either significantly reduced the virus titer or completely cleared it prior to infection, and there was no evidence of immune pathology. The gray is that there is no sign, no difference in vaccinated and mock vaccinated animals, so the vaccine didn't really work, but it didn't make anything worse in terms of weight loss. And red is examples of animals that had extreme increases in weight loss and or death. When you look at inflammatory disease in the lungs of these animals, you can see that the number of red squares increase significantly and the number of blue squares are reduced significantly. So as you begin to move vaccine outcomes in genetic reference populations of natural outbred populations, adverse reactions may become more prominent, and that needs to be thought about in the context of vaccine development. Next slide. So with that, I just acknowledge the people in my lab and thank the working group that put this important meeting together for inviting me to talk.